Welcome everybody to the first Nanome live stream of 2023. I was just talking about how back in the day we did some live streams on YouTube and even a few Twitch streams back in the day. And the last couple of years we've done webinars definitely check out nanome.ai/webinars for webinars. We got a lot of great reception and feedback. And one of the things that our webinar audience and our YouTube audience have wanted to see is more actual live usage of Nanome. So we decided to dust off our streaming computer and organize this stream. Today, we've got our very own application scientist, Roberta Bartolotta, joining us from Basel, Switzerland, as well as our CEO, Steve McCloskey, right next to me here in San Diego. So these are just the slides I was just showing earlier. Roberta has over 10 years of international experience in both academic institutions and biotech in the field of anti-cancer, antiviral research, and new modalities. She has a PhD in pharmaceutical sciences, medicinal chemistry. She contributed to advancing several medicinal chemistry and drug discovery projects from early stage hit finding, lead optimization to candidate selection at Selvita and SpiralChem. Roberta played a key role in adopting Nanome at Novartis Institute for Biomedical Research in Basel before joining Nanome as an application scientist. Today, we're going to be analyzing the crystal structure of human cereblon. Give me just a second here. Let me switch our slides. There we go. The unique thing about this stream is that it'll all be in the new Quest Pro and its Passthrough Mixed Reality feature. So for those of you unfamiliar with Passthrough Mixed Reality, it's more akin to augmented reality, where it feels like hologram models of molecules and proteins that are on top of your real world, as opposed to transporting you to a whole new world that virtual reality does. With mixed and augmented reality, much of Nanome's value propositions from virtual reality is still there, while also being able to naturally navigate the real world around you. Now, last housekeeping note here, as you've already started to use the comment section, we will have a Q&A section toward the end of the video. So feel free to put your questions in the YouTube chat box, which you should see again towards the right of the live stream on YouTube. So yeah, without further ado, Steve, I'll let you take it from here.
Okay, great. Thank you, Keita. So yeah, I'm actually joining live from the Nanome office. What you are seeing is actually what I see in the headset here. So we have, Keita with the streaming computer setup. You can see that I have my avatar hands kind of overlaid on my hands to help point things out. Then over here, we actually have Roberta joining live from Basel, Switzerland. How do you like telecommuting to work like this?
That's amazing. This is the very first presentation that I'm giving in VR. I'm used to just laser pointing at 2D screens, it's amazing.
Yeah, fantastic. All right, so we have the Human Cereblon right here. Do you want to introduce some of this protein and tell us why this is a really important target right now for drug discovery?
Yeah, sure. In a minute, it will be loaded even here. But in the meantime, we're going to talk about the Human Cereblon E3 ubiquitin ligase complex, which is the target of approved drugs as lenalidomide, thalidomide, and pomalidomide, which are really very important in the cure of multiple myeloma and B-cell malignancies. So these drugs actually are useful to promote the recruitment of substrates as high-chiros and aiolos for further ubiquitination and degradation. We're going to show the very interesting part of this protein. We know that it's constituted by the DDB1, which is the blue part, and the thalidomide binding pocket, which is the C-terminal part here around. And we can see that far from this domain here, we have the zinc coordinated by 4 cysteines.
Yep, it's going to be right up here. Yep.
Yeah.
We have our zinc coordinated by our 4 cysteines with the sulfurs colored in yellow right there.
Yeah,
Just give a nice
exactly.
view.
And they should be distant from the thalidomide binding domain around 18, 20 angstrom. You can even measure
Yeah.
that.
So yeah, let's go ahead and use the measurement tool. And so I went ahead and changed it over, a little hand measurement tool. And we're just going to overlay that on the zinc as our starting point. You see that highlighted in yellow. And then I'm just going to select about right here. And we can see that that's, yeah, about 19 angstroms. So pretty much, yeah, what you said, 18, 19 angstroms distance between the zinc and our binding domain over here for the thalidomide.
Yeah, actually, the binding pocket of the thalidomide is very well featured by 4 tryptophan residues, the 380, 386, and the 400, which confers the aromaticity of the binding pocket here, and so the hydrophobic feature of the binding pocket together with the 4 tyrosine here over there,
Yeah.
and the
Here,
phenylalanine. Yeah.
Yeah, I'll go ahead and highlight those. So we have our 3 tryptophans. So we got 1, 2, and then 3. So it's going to be 380, 386, and number 400. And then you said our which tyrosine? It was the 355.
Yeah.
Great. And so I'll just go ahead and select all those. And I'm going to just change the color just to be a slightly lighter shade of pink, just so that we could help differentiate that a little bit better. Great. And so we can see in the light pink, these are kind of the more important residues that actually build that pocket out, as well as this really important residue that helps kind of keep the structure of the binding pocket intact. But yeah, maybe to just take a step back. So really important protein complex here, but since we're just going to be focusing on the pink portion, I'm going to just go ahead and get rid of the blue portion so that it doesn't look too cluttered for our viewers at home. So yeah, I will go to our hierarchy menu, select chain A, as well as the heteroatoms of chain A, and I'm just going to delete them. I could also hide them, but since we're just going to be dealing with the pink part of the protein, I think that's just a little bit easier to get rid of that for now. And so, yeah, do you want to explain, why people are still targeting, this binding pocket and trying to come up with new drugs, even though there's already available treatments on the market?
Absolutely, Steve. The fact is that these FDA-approved drugs present teratogenic effects, side effects. So that's why there is a huge effort by researchers to find analogs without any side effect. And still, they keep as a main feature the glutaramide, which is the part that is involved in the interaction with the tryptophan residues, and exactly the glutaramide, the nitrogen of the new glutaramide forms hydrogen bond with the histidine here that you can see, both with the imidazole and the side chain. So this is a very, very important portion, is the pharmacophore of immunomodulators compounds such as the lenalidomide. So when the researchers put an effort to find the new analogs, actually they try to keep this portion and just changing the isometalone part. And yeah, we know that in the thalidomide, we have a carbonyl here this way, and we have a thalidomide core. But yeah, we can even search more publications right there, and we can try even to make new analogs just using our medcam tools. But before I would like also to highlight some important features, because, for example, the the amine group of the aniline part of the isotonium core doesn't interact, yes, with the glutamine here, but actually the distance is very huge. So that's why it's not really very important. We can even try to not keep this moiety and just apply some structure modification to these parts, for example.
Yeah. Let's go ahead and kind of identify a few, good, hydrogen bonds that are probably forming between this oxygen and this nitrogen, and then this hydrogen on that nitrogen and this oxygen. So we could see that, those are probably forming some really good hydrogen bond interactions. But measuring it is one thing. I think it'd be better if we actually use one of our plugins to calculate the chemical interactions.
Definitely, yes, Steve. But may I ask you, please, just in case, I just go out the room and I re-enter, or you may reload the protein in the meantime, please.
I think that's fine.
Thank you.
So I'll go ahead and run Nanome stacks, and I'm just going to pull up the chemical interactions plugin. So the chemical interaction plugin allows you to calculate and visualize chemical interactions made between proteins and small molecules or other proteins. And when I click run here, I'm going to have some options on what type of interactions I want to show. I'm just going to change the pi aromatic to just appear in a, let's call it a white, just so it doesn't interfere too much with the magenta that we already have there. And now I'm just going to click calculate. Cool. And that's just going to go ahead and load some interactions into the workspace. In the meantime, I'm actually going to get rid of those measurements I made by using the measurement tool and just deleting all of those right there. So we could already see that there are some of those pi interactions happening between our ligand and this histidine 353, as well as some hydrogen bonds forming really deep in the pocket over there as well. And you really get a sense, when you get into this pocket, just how much these tryptophans are holding in this ligand and just creating a really nice pocket there for our ligand to fit into.
Exactly. Yeah. Thank you, Steve. I'm back here. So what I wanted to show you before exactly is the distance between this glutamine here and the aniline here, the nitrogen of the amino group of this portion here. Looks like being really very distant to make any stable interaction with the ligand.
Yeah, great. I'm just gonna get the menus a little bit repositioned here, and looks like you've joined at a different part of the office now, which is perfectly fine.
Just went around.
Yeah. So what were you just saying a second ago?
Exactly. Nicely, Harry has shown the hydrophobic interactions we have seen between the tryptophan and the pharmacophore, the glutamide, just summing up these residues and this tryptophan here. And it's very well known that any mutation of these residues, the tryptophan 386, may bring it to affect the affinity with the immunomodulators compounds here. And
I'm just going to generate a few more interactions, really within that, the pocket to see the interactions between the various residues, because it really does help stabilize the entire pocket, even though some of these residues aren't actually interacting. So, for instance, this histidine 397 looks like we have a good interaction pi network between the tryptophan and really this other tryptophan, and this just all these white lines basically represent the stability that's being offered by the various residues surrounding the pocket.
Yeah, I can see very well here. The phenylalanine doesn't take a part of the interactions between the ligand, but just gives stability to the protein. Here, you can see the interaction between phenylalanine 402 and the phenylalanine 381 here.
Great.
Yeah, but the glutaramide is the pharmacophore part, but as in the part is the solvent exposed portion of the immunomodulators compound. And we can even detect these just showing the surface around the ligand.
Yeah, so I think that'll be good to just kind of clean up some of the interactions there, and then actually use a different plugin to generate a high quality surface.
Yeah.
And we'll just select our protein 4TZ4, chain C, and then go ahead and click generate.
Yep.
And this is going to just generate a nice surface for us in a few seconds here. And then we should see a really nice pocket. Yeah, we could already see that forming. So yeah, in yellow, we have our protein surface. And if we really just zoom into this, we could kind of see that there's a really nice amount of space that this portion of the ligand is occupying. And then, as you said, this portion over here is pointing out to solvent and not interacting as much with the residues, especially getting all of those hydrophobic interactions between the tryptophans in there.
Exactly. This is exactly the portion that promotes the recruitment to substrates as iCarriers and alleles to further ubiquitination and ubiquitination and degradation. In this way, we can really see where we have still room for structural modifications. So why we do not go ahead, then we can just make some.
Yeah, here I'm going to actually turn off some of those labels. They're poking through the surface a little bit. There we go. And let's also change the color of our surface. This yellow is nice, but I think because of the protein being in magenta earlier, yeah, this magenta color looks a little bit nicer in terms of consistency. So very interesting protein structure all the way around. But you can see that there is a really nice and deep pocket right there in the binding domain.
Yes, nice.
Yep.
What we can do actually is already assign a different color to this ligand, maybe duplicate and make some analogs. What do you think?
Yeah, that sounds great. So let's just go ahead and kind of reposition ourselves around the office here. Roberta, if you want to kind of go to my right there, I think that'd be great. So I'm going to select our ligand. You can see that's been selected, and I'm just going to use the modify menu to duplicate it. And so now this brings up basically a copy of our ligand right here. And we're going to be using this copy to do a few modifications based actually upon this paper. So really quick, shout out to the authors of this paper, Mikhail Krasavin, and everyone else that has taken part of this. This is from the synthesis of novel glutaramide ligands for the E3 ligase substrate receptor Cereblon, which we just saw there. An investigation of their binding modes and anti-proliferative effects against myeloma cell lines. And if we go a few pages in to this PDF, and you can load any PDF into Nanome, it's really great to actually see some of these compounds just right there at the bottom as a reference so that we could draw them. So how about I'll go ahead and get started on modifying this into compound 8. So first I like kind of lining it up and getting most of the structure kind of how it was. And then we could start making some modifications. So I'm going to go to the MedChem tool. I'll start out with the deletion tool. I'm going to delete this oxygen and that carbon. And now I'm going to actually just build that into a 6-member ring. So add one more carbon and then connect them. Okay. All right, so now we can see that we have the 2 6-member rings. We're gonna change that into a nitrogen right there. Put the oxygen coming off right at the top there. Make sure it's double bonded. Okay, and we have another double bond right there. Great. And then it looks like the rest of this is correct. And as a final step, I'm just going to go ahead and add some hydrogens right here as well. Okay, cool. All right, Roberta, what do you think? Did I make that compound? Oh, actually, I'm missing one carbon.
There's the methyl group here missing, exactly. So we actually work to from the azindorinone core to the quinazoline compound here. And yeah, all the hydrogens are also shown over there.
And and I'm also going to change the coloring on this as well using our color menu. And let's go ahead and make this one into this nice kind of yellow color right there. So the carbons now are going to be yellow. We'll keep the nitrogens and the oxygens in their traditional blue and red colors. But yeah, Roberta, yeah, did you want to take a crack at designing compound number 11 with your MedChem tool?
Oh yeah, absolutely. So
I'll give you presenter there.
thank you so much. Yeah, that's very nice to do that. So we remake basically the glutamide here, which is, as we said, is the pharmacophore of these immunomodulated compounds. And they're gonna have here, oxygen here, nitrogen here. Yeah, I really like this spiral cycle. It's nice that it's minimizing while building.
Mm-hmm.
Yeah, I just need one more nitrogen here. And then sulfur.
Great. Yeah, this is looking like a really nice compound. It's shaping up, coming together really well.
Kind of manufactured to sparkling compound. And then we have here. Yeah.
Okay.
Like,
yeah, that's looking fantastic.
what do you think about this?
Cool. Yeah. So yeah, let's go ahead and yeah, check it out. So we got let's see, that group down there. That makes sense. Sulfur, nitrogen, and yeah, the OH. So perfect. And yeah, of course we could add some hydrogens to that as well. Perfect.
Yeah.
All right. And then actually, since we just made that, we'll go ahead and color this one a different color as well. Let's go ahead and pick this nice cyan color. Great. So yeah, we have compound number 8 over here in the yellow, number 11 in the teal, and then of course we still have our original ligand still docked in our protein already. Cool. So yeah, we don't need to have this PDF so large anymore. We just go ahead and place that over to the side. If we get our protein structure just back over here, we can actually see that our original compound is still there in the green. And if we select everything, we should be able to just go ahead and realign everything as well. Or what would be even better is if we actually use the docking tool. So yeah, we could see that we realigned the yellow structure that we modified and the green structure. It's relatively in there. And then this new compound, yeah, we're gonna have to either manually dock or use a docking tool, which I think will be a little bit better, so that we're not kind of guessing exactly which pose it would be. but we could maybe get several different poses and calculate the energy scores of each one. Great. So first off though, I'm going to hide the surface here that we have, just so we could see some of the surrounding residues again. So I'll just go ahead and turn that off. Great. And so now we could really see how these ligands would overlap and still be interacting, really deep in that pocket. We keep a lot of these same interactions that are kind of buried in there, but then we've mostly changed some of the bits over on this side of the molecule facing towards solvent. Cool.
Yeah, I'm assuming that we are keeping the same hydrophobic interactions as before because we kept the glutamate part.
Exactly, yeah. So that glutamate part should still be interacting with our tryptophans really deep in that pocket. And then over here, hopefully, these additions have less side effects, right? So the goal of doing some medicinal chemistry with these motifs. So we could calculate the docking scores. So let's go ahead and do that. So just so that we have a baseline as well, I'm going to actually split out the original ligand so that we can also redock that and see how that gets posed. And I'll just lock those so we don't move them out of the way. All right, so another plugin that is really great to use is going to be the SMENA docking plugin. So if we run that, we're going to have several options here. So to start, we're going to be selecting our ligands. And so our ligands are going to be the original ligand and the two that we made. So I will just go ahead and select 4TZ4, the molecule we made, and 4TZ4A, which was our original. Our receptor is going to be this 4TZ4, which was the protein. So that's going to be in pink there. Then the last part is going to be the site that we wanted to look at. And I'm just going to choose our 4TZ4A, which is right in the pocket. And you can see that now we have this sphere really right around that area. And so this is where Autodoc SMENA is going to be looking to dock the new compounds. And we'll just do 2 poses for each compound and then go ahead and run it. So right now it's spinning up a cloud server. We are basically exporting all of these molecules in a traditional type of structural data file. That is going into Autodoc SMENA with this protein as our receptor. And then it's running the docking calculations to see which poses they might take and what energy scores would be associated with those poses. And we could see that it's already been loaded back in right now. Great. And so, I could get rid of that menu. And so to start off, let's go ahead and change the display to be a nice wire view, just so we could see each one a little bit better as they overlap. Additionally, we could change the colors. And so our 4TZ4 original compound, so this modified one was yellow. So I will keep with that same structure and actually make this one yellow, maybe a different shade of yellow, just to show that there's some difference there. Really saturate that yellow. Our next compound, the molecule, I believe this one was the blue, that's cyan. And so the docked molecule, we'll go ahead and make that into a cyan as well. And then finally, our original ligand was in the green, so we'll make that green. And so now we could see that we have all of our compounds similar to their original colors. We could see that we have the scores a little bit overlaid right now, but we could also pull up the menu to analyze the scores, and we could switch between the 2 poses as well. Great. So we have 4TZ4A, our molecule, and our 4TZ41. So we could see that we got a minimized affinity of negative 9.139. That was on our the first modified compound that I made, the compound 8. The original compound got a minimized affinity of negative 9.46, so a little bit lower, which is good. And then the third compound that Roberta drew is actually probably the lowest minimized affinity. You could see there at the negative 7.73. But you can see each one of these had 2 poses. So you can see in the gray there, that is the secondary pose, a little bit higher minimized affinity. Same here, higher minimized affinity and higher minimized affinity. But it's nice to, kind of compare, the multiple poses and kind of cycle through them using the playthrough buttons that we have there. Great.
Nice results that can be also ranked. And we can even compare with the physical chemical properties of the 3 compounds.
Yeah, that sounds good. So I'm going to go ahead and we'll keep those there. But let's use the Nanome stacks now to actually run through and do a chemical property calculation. And so this is using RGKit to calculate the chemical properties of the ligands. But of course, all these plugins are modular, and so if you want to do something else, you could just actually swap out RDKit and use a different chemical interaction calculation software as well. So I'm going to take our 4TZ4 compound I drew. We could see that these are its molecular properties: molecular weight, log P, total polar surface area, estimated solubility, hydrogen bond acceptors and donors, as well as rotatable bonds and aromatic rings. But all this data should be consistent across our molecules. So we'll just take a snapshot of that. Go to Roberta's compound, we'll take a snapshot of that. And then we'll go back to the original ligand and take a snapshot of that. And so now we could actually view all of the data side by side here, which is really nice to see, just some of the big changes, like, really log P really changed drastically between these compounds, while molecular weight didn't change too much.
Yeah, can we make it also even a little bit larger, the window maybe?
Yeah, sure. Yeah,
Okay.
we could make that a bit bigger for the guests at home.
Yes, it's very good. For you have a good overview of the all molecular properties compared here, and you can even export the CSV, and you can use documents, including documents.
Yeah, so we could export all this as a CSV, so we could use it with other software, or just kind of looking at these in a table format as well. I'm going to unlock our original compound there, just so I could pull that out and do an overlay to show you which compounds were which. So we have our original ligand right here. We have the compound that Roberta drew, right? So you get the right orientation, pretty much like that. And then we have the modified compound that I made. Let's see. Yeah, I think that orientation is about right. Cool. Yeah, so you get all the chemical properties alongside all the 3-dimensional ligands. And of course, all of them are inside of the protein structure here, as we can see by these overlaid wire structures in the binding pocket.
So Steve, we're coming about the 20-minute mark here. We have a bunch of questions. Is there anything else that you want to share before we move on to Q&A?
No, I think it's great to get started with some of the Q&A. Let's go ahead and move on to some of the questions here.
Perfect. So yeah, earlier it looks like Roberta was having some issues with the presenter. So we got some comments about essentially like, well, I think they were correct in the sense that Roberta did have some issues, but now those are been resolved. But they did want to get a better idea about kind of the presenter mode and how that works.
Yeah, so right now I'm the presenter, so I could grab and move everything. Everything's locked together in relation to this protein, but we can also actually select all and toggle the lock to unlock everything, and that's going to allow us to move everything independently. And what we could do here is actually I can give presenter to Roberta, and then Roberta could actually, grab and interact with these protein structures and molecules as well.
Yeah, sure. And there is also another way I can even steal the presenter if I want, just with the action menu. And I go to room and I steal the presenter here. See, I can even give the presenter to other guests here in the room.
Yep. Yeah. So just on my menu, yeah. So I could take presenter back, but it's just one at a time, so you could switch it. Also, there's a quick shortcut, and so I just reached out and grabbed it without stealing it. So this is called a grab to switch presenter, and so if Roberta grabs it now, you can see that her crown has changed to presenter mode. And yeah, this is a toggleable item, so that you basically go into the room menu and you toggle on allow grab to steal presenter. And so instead of clicking every time I wanted to take presenter, this steal presenter button, I could just wait for it to let go and then grab it. And now I'm the presenter and can make different modifications and measurements.
Yep.
Cool.
Perfect. Awesome. We got a bunch of questions on one in particular about any plans to integrate large language models such as ChatGPT. Can you use voice commands to accelerate the workflow and the way you do selections, change colors, visualizations, and request basic analysis?
Yeah, that's actually a really, really great question. So right now on Windows 10, we've integrated the Windows 10 API for voice commands. And so if you are actually connected to a Windows 10 PC with your VR headset, you can actually spin up voice commands. We have a list of short voice commands such as, select all, color blue, hide chain A, different sort of shortcuts like that. Unfortunately, we have not brought that capability to standalone devices yet. I do believe that in the future, it basically integrating it with Nanome stacks and having this all be a cloud API as basically a voice command that you could use is what we're moving towards. Additionally, we also have the macro system. So the macro system allows you to pre-compile different types of views. And so if you do like a site view, when we run this, it should actually automatically calculate where the ligands are, and then basically do a angstrom selection around that. So we can see that it picked up the zinc as a ligand, the one that we talked about that was earlier complex beta-cysteines. And so this macro will actually select all the surrounding residues from any heteroatoms, because I did site view all. And so anything recognized as a heteroatom is going to be our input. And then it's going to generate the wire view as well as the labels, and then the molecular surface in a semi-transparency, as you could probably see there on my screen. This is just one example of a macro. And so our philosophy is if you combine voice commands with plugins, so you could say, hey, doc this for me, or calculate the properties instead of clicking the buttons, combining that with the macro and scripting capability, you should be able to eventually, have that full Iron Man Jarvis type of interaction. Definitely moving towards that, and great question. Appreciate you asking that.
Nice. How can we export the model and, for example, perform external calculations?
So right now what we're doing is basically exporting this as a PDB and this is SDF in order to perform these external calculations. But let's just say you had a longer simulation like molecular dynamics. What you could do is actually select what you want, and then we could go to this button right here. It says export. And now we have the option to save it. We could just label this for TZ4 new. We could save that in the PDB format, SDF, MMSIF, or SMILES. So PDB is pretty common. I'll just export that. You could do this locally, or you could also upload this to the cloud storage plugin, which we call Vault. And so I'll just put that into the Vault structures of mine, export that. And now that's going to be available as a PDB format. And so you could do the same. You could export these as SDFs. But yeah, pretty much anything you make and modify. If you mutated residues, if we change some of the atoms here, you could save those as the traditional file formats that you would typically work with other programs with.
What other tools do you have besides measure, selection, and medchem?
Yeah, so there's actually several tools. I have the quick select tool menu right here to go through them. So the hand is actually a great tool in of itself, allows you to grab and move things. Of course, that capability is going to be consistent, where you could use the middle finger to grab across all the tools. But the main difference is going to be how the index finger is used. So the measurement tool allows you to make measurements, move them, make angle measurements. So you could actually measure the angle between 3 molecules, or atoms. So you can see this nitrogen, carbon, and carbon. We can see that it's an angle of 118.4. You could also do dihedral measurements in case you wanted to do 1, 2, 3, 4. Oops, I think I misaligned that by 1, and distance as well. And so I'm just gonna clean those up with the delete all functionality. Additionally, the selector tool allows you to do residues, chains, molecules, arrange, or individual atoms. If you just wanted to select an individual atom, you can do that. The torsion tool is really fun because it actually allows you to twist around rotatable bonds. So you just kind of go in there and then use your index finger, and then it's like twisting a doorknob. So pretty intuitive. The medchem tool, which we used earlier, allows you to build, edit, and delete. Actually has full periodic table access in case you wanted to build with some more exotic molecules than the ones that we include on the quick menu right there. You go ahead and just replace that. Pretty straightforward. And then the last tool is actually the drawing tool. And the drawing tool is really great for collaborative activities where maybe you just wanted to make some quick notes and say like I don't know, compound 1, 2, and 3, and just kind of I don't know, get little bits of data to say like that was good, that one was bad, and this one was like really good. There could just be various collaborative portions. You want to make a note, you want to write an agenda, just generally sharing information on a whiteboard. And of course, you could save this, export it as an image, and you could also mark up images and PDFs as well. In case we actually wanted to do that, we could take this PDF right here, use the drawing tool, and that should allow us to now mark up our PDF and highlight, as well as erase things that we might have drawn by mistake.
So Steve, Zorn's actually in the chat, watching us right now. He asks, is there a way to export Conformer generator or Semina docking energy and RMSC data output, or output data numbers for multiple conformers in some kind of tabular CSV form?
So we do have the Conformer generator tool. So that's going to be over here. So we can generate conformers using a plugin. So if we open that, we could select our entry. So I'm just going to pick this one right here. We'll just get that close. Great. And so we're going to generate several different conformations for this. Might not be too many. It's not really too many rotatable bonds. And if we click generate, that'll go ahead and start generating a 100 conformers for this, as we can see selected there. And that might take a minute just because it's generating 100 of them. So it takes a little bit of time. But yeah, once we have this generated, we could actually use the data table plugin. And so this data table plugin allows us to use the web browser. So this is actually a general purpose web browser. It allows you to go to really any website. You could go to our documentation page, you go to YouTube and watch some of our tutorials, our CSV protein data bank. These are just shortcut tabs, but you can just input really any URL into the browser up here. But what we've done is we've actually made a web browser plugin compatibility where now when I run this data table plugin, my various molecules. So just maybe while we're waiting for the conformations to generate, we can see that we have our minimized affinity of our two dot compounds in this table view. We have various settings as well as the capability to graph this. And then additionally, if we were to use the chemical properties plugin again that we had earlier, you can actually view all these snapshots of your various ligands and residues, and you could export all of that in the common CSV format by clicking on this export CSV.
Diego asks, can you talk about how one can save visualizations of viewpoints to produce tutorials?
Yeah, so let me clean up these menus a little bit. Just put them in the background there. So there's different ways to save this entire workspace. And so what you could do is you could actually save this as a Nanome session. And when you save as a Nanome session, it's going to take all the coloring, all the representations, and it's going to just save that as is. And so earlier what we did was the export functionality, but now I'm just going to use the save functionality. And so when I click on this save functionality, so I'm just going to put these in my middle grounds, get that a little bit closer. So we could save the workspace, and we could just name this, let's call it 4TZ4 new. Cool, and it's going to be in a .nanome file format. And so this .nanome file format actually saves the representation, the coloring, and all that information. And again, I'm just going to put this into my Vault workspaces here and save that. And so now, all the position of the molecules, all the representation, all of that has been saved. And so instead of exporting a single protein as a PDB, we've now exported the entire workspace as a .nanome file.
Great. Do you want to talk about spatial recordings a little bit?
Yeah, okay. So I guess the question also involved tutorial making. And so what we've done is actually created a content discovery portal, which allows you to make and play spatial recordings. So spatial recordings are basically, as you see my avatar and Roberta's avatar. Here, maybe I'll use the camera tool to kind of give a better idea of what that looks like.
You can see the viewpoint.
Yeah. So yeah, on the camera, unfortunately, we don't have the mixed reality enabled, but we can actually see our avatars in virtual reality. And so what the spatial recording looks like would be like recording a video of us, interacting with the protein and such, and kind of explaining concepts. This is actually saved as a full three-dimensional recording. So we call it spatial recording because it records the avatar movement, the voice, and all the protein movement and modifications. And what we've done is put together a content series, which includes an intro to Nanome, some of the capabilities, as well as some lessons about chemistry if you're interested in learning more. And then some tutorials on how to use the tools and other features within Nanome, including more complex ones like the chemical interactions and designing small molecules with the MedChem tool. And yeah, when you play these, you'll have the avatars kind of, playing through the recording and showing information. And of course, I could actually make my own as well using this spatial recording button. And so basically I would just hit this button and then we would record the session.
Another question from Diego asks, a lab mate run multiple projects. How can we, a principal investigator, follow these different projects and files?
Yeah, so I think that Vault is actually a pretty good place to do that right now. This is something that we're constantly improving, but you have this option for the My Org functionality, as well as your individual account. So on the individual account, you could have all your recording structures and workspaces saved. We could see that there was that PDB that we exported earlier, workspaces. We have different workspaces here as well. And these are all part of my account. But again, you could use my org, and then that could be shared within your org, which could just be your research group. And so you would see Other people could make folders, and you could kind of check in on the structures through the vault.
Did we go over exporting models for performing external calculations?
Yeah. So earlier I had actually used the export to export this 4tz4new.pdb. And if we were to actually load that in the session, it's just going to be kind of a blank copy of this protein right here. So I exported this protein, but when you do PDB, it does not save any of the representation. And so when we import it, we just get sort of the default cartoon coloring when you import any regular PDB. But if you do save this as a workspace, a .nanome file, you will retain the coloring and all the representations.
Another question, is there a way to see the sequence of a protein? What are capabilities related to the protein sequence?
Yeah, so definitely is a way to see the protein sequence. So actually that new one looked a bit cleaner. Let's do that. So we'll zoom in, and then there's two ways. So we could use the hierarchy menu to actually go in and look at the sequence in more of a grid view. And this could be good for just kind of looking at all the numbers in a really quick way. But what I like to do is actually use the sequence menu. And so this sequence menu will allow us to select our structure and our chain, and then it'll sequentially give you the entire series of residues right here. And we could also select individual residues and highlight them on the structure from this menu, as well as from the other menu. So you could select different a whole bunch, and you can see now that this alpha helix kind of has a little bit of a green highlight, which represents all these residues that we just highlighted there. And of course, we could see that that's consistent across the sequence menu, where you can see that we've selected this row of amino acids in our sequence.
Another question, can you do 2D chemical drawing?
2D chemical drawing, yeah. So I'd say our strong suite is three-dimensional. Obviously this is like the only professional virtual reality application for three-dimensional molecule building. But we do have the capability to interact with web apps. So if I pull our web browser up, we could pull up this MolView 2D drawer. We'll just go ahead and disconnect from the earlier menu that we had there. So we can see that this is actually a two-dimensional compound that we're interacting with. Kind of have my cursor as like a mouse. So I'm just turning that in the web browser. And then of course, we could actually make modifications on this 2D web app. So yeah, I mean, I'm a bigger fan of three-dimensional drawing than I am with two-dimensional drawing there. But you could see that those modifications I made were just actually made onto the molecule. And then what we could do is actually leave tools, and then we go to a mol file. We could just download that and have the structure in 3D right in front of us. And so this is a nice way to actually interact with web apps, where you could do drawing in 2D over here. You could see the three-dimensional preview, kind of rotate it. It's still flat in 2D for me, but then we could just download it and it becomes a real true three-dimensional molecule. And of course, if we wanted to make subsequent modification on that, we could do that relatively easily as well.
I think those are pretty much all the questions. I think last one quick technical question. If you could talk about the mixed reality capabilities, how to toggle between VR and MR, and then just talk about the hardware.
Yeah, so I'm just going to close all of our menus here. Let's go ahead and make these structures a little bit smaller. All right, so yeah, right now I am at the Nanome office. Again, Roberta is joined with her avatar. Of course, Kata is helping with the stream over there. Mixed reality is pretty fun because you could, of course, see the world around you. Like I know that I'm not going to bump into this chair, and I could interact with that. You could do things like, place your protein inside of your office and kind of, have fun with spatially located proteins. But then switching between mixed reality and virtual reality, it's actually just clicking on this button right here that says mixed reality. And so you can see now this is the pure virtual reality environment of Nanome. Of course, you could change the sky sphere. You could hide the floor as well. But yeah, you could have your protein structure here. Maybe it looks a little bit better with this contrast, or if you wanted a complete dark environment, you could even do that. So if we wanted sort of a full contrast, just be in a black or a white session, maybe this helps with different taking pictures. But of course, I like the sunset, and even more than the sunset, I like mixed reality, because then I could actually see the real sun outside of our window a little bit.
We just actually got another question, so wanted to just address this one. Can you run MD simulations inside of Nano?
Yeah, so what you saw earlier with the molecular building, and I'll just kind of do a really quick one right here. And so we have a local energy minimization. So if you were to do something like, maybe draw this oxygen just pretty far, we could see that the minimization plugin actually corrects that. So that is using force fields to calculate that. But again, it's a really local action.
I think we ran out of sorry, go ahead.
Yeah. So what we're gonna show is actually importing a molecular dynamics session. So after you have ran the calculation, you could actually import the trajectories and then analyze them. And so sometimes it takes hours to run these calculations, or even days if they're really big. But what we could do is just play them back using that same multi-frame menu that we were using earlier. And of course, we could pause it and see different frames, do things like make measurements, maybe just between distance right there and right there. And then when we play it, it'll actually be updating across them. So this is a really great platform for analyzing molecular dynamics. And if you did want to make changes like modify residue, export that, you could actually export this in PDB or MMSIF to rerun molecular dynamics, calculate that, and then analyze it again.
Cool. So I think that's pretty much it. And we can probably wrap up here. But we do have one kind of last question. I promise this is the last question, which is talking about the perform customized actions, talking about plugin development. So maybe, Steve, if you want to just give a high-level overview of Nanome Stacks, what it's like to make plugins, things like that, and then we can wrap up.
Okay, great. Yeah, so the question was about plugin development. And so, Nanome Stacks is our plugin platform here. So you can see that we've organized these into different types of stacks. So you could see, if you wanted to just look at the computations or the alignment ones, like superimposing an RMSD or coordinate alignment, or just look at all the plugins. So some of these are made in-house, like the superimpose plugin was a really nice one that we made. But there's also third-party ones like Smina docking is Autodoc Smina. But I'd say in general, it's all Python based. And so what we provide on our documentation page is actually a little bit more information on all this. And so if we look at the documentation tab there, we could see Nanome Stacks. So this is docs.nanome.ai. And then you could go to the overview. And this gives more information about the capabilities. But yeah, you would pretty much just be doing a little bit of Python scripting to either connect tools that are already available and running, or you could even just make your own Python-based algorithms and integrate directly with Nanome there. Yep. And yeah, of course, you could learn more about creating your own, which will take you to the more in-depth documentation page, which explains a lot more about the architecture of the API, the plugin API itself, and gets a little bit more technical. So I'd say if you're very serious about actually developing plugins, then go to docs.nanome.ai and then go to this Nanome Read the Docs page and learn more.
Fantastic. Well, thanks everybody for tuning into today's stream. We love having you all, and we're trying to do more of these. So please feel free, or we'll see you probably in the next stream. So thanks, Steve and Roberta as well.
Cool. Yeah.
Thank
Thanks
you.
everyone for tuning in, watching, and thanks Roberta and Kata for helping with this stream, making it really nice.
Yeah. Thank you very much.
All right. Thanks everyone.